Afternoon, everyone. Welcome. We'll be starting the webinar in a couple of minutes. We're just waiting, giving a bit more time to allow some more people to join. Hello, thanks for joining us. We're just waiting to allow some more people to join, and we'll be starting shortly. Afternoon, everyone. We're just waiting for more people to join. Please, could you just put any technical queries that you might have in the Q&A as opposed to using the chat function, which should be disabled, or raising your hand. The Q&A function will also be used for any questions you might have later on in the webinar. Okay, I think it's time to start. Welcome and thank you for joining us for our inaugural webinar with Dr. Gavin Arno, who is joining us today from South Carolina, which is on the decoding the genetics of inherited retinal diseases. And it's the first in our series of Fight for Sight presents. So before we get started, I'd like to run through a couple of housekeeping rules. If you'd like to, live captions are available during the webinar, and we will be using illustrated slides to guide us, but we will read out everything on these. If you'd like to revisit the webinar later, or you have some friends, colleagues, family who intended to come along to the webinar but couldn't make it, the slides, a transcript and a recording will be made available to you post-webinar, so you don't have to worry about taking notes. As I mentioned before we started, the chat function has been disabled because it's distracting for those using a screen reader, and it's because it's simpler for us to monitor one channel, which will be the Q&A window. So please use the Q&A window to ask any questions that you might have during the webinar for Gavin later on during the Q&A session. And I just wanted to give you forewarning that when we end the webinar, it may appear to end abruptly. This is perfectly normal, so don't be alarmed. And we will try and give you advance notice of this. So on to what to expect. Well, first of all, we will have a welcome from me, Emma Blamont, Head of Research and Programmes, and an overview of our research funding offer. We will be introducing Dr. Gavin Arno and his research. Then we'll move on to a live Q&A session. So please type any questions that you might have in the chat function, and then we'll wrap up. And I'll explain about how the resources will be made available to you post-webinar. Okay, so on to me, your host. My name's Emma Blamont, and I'm Head of Research and Programmes at Fight for Sight. I used to be a lab scientist back in the day, an immunologist specifically. And since leaving the lab, I've worked within other funders like Breast Cancer Now, the National Institute of Health and Research, and Scleroderma and Raynaud's UK. And I'm really happy to be at Fight for Sight and working with the research and social change programmes in sort of advancing the great work that we do and in hosting this webinar and sort of allowing people to discover more about the work or learn more about the work that Gavin has done. So at Fight for Sight, we fund the brilliant minds and the bright ideas that put the change into sight. And for anyone impacted by vision loss. And we do this through investing in sustainable projects that deliver equity and fund science that will help us better understand, diagnose, prevent and treat vision loss. So this slide shows one of our researchers, Dr. Matteo Rizzi from UCL, at work analysing some images that have come out of his research. So Dr. Rizzi and his colleague, Professor Omar Maru, are undertaking research into Stargardt disease, which is an inherited eye condition, and they received a project grant to do this from Fight for Sight last year. So our research funding has a really important place in the research ecosystem. Research into preventing and treating sight loss is chronically underfunded and receives a mere 1.2% of publicly funded health grants. The researchers that we fund really are at the cutting edge, making breakthroughs and discoveries that will help us better understand, diagnose and prevent and treat eye disease. So we're not just a research funder, we also fund projects that can help drive social change for people who are blind or visually impaired. And this is an example of one of our social change projects, which features Jamie's Farm in Hereford. And the picture on the slide shows a group of young vision impaired people sitting together around a kitchen table with Dave, the head farmer at Jamie's Farm. And this was part of our loneliness and isolation funding, where we funded farm visits for young people to experience the benefits of being in nature and build connections between each other. So these partnerships are really pivotal for changing the lives of blind and vision impaired people. Research shows that people who are blind or vision impaired are 3 times more likely to experience loneliness and isolation than the general population, which makes these type of projects really rewarding for us as a charity. So this brings us on to our research strategy. So we don't just invest in projects, we invest in people, and we aspire to build a vibrant community of vision researchers, foster collaboration and accelerate partnership and progress. And the diagram on this slide really represents this. And at the centre of our diagram, and as our activities and organisation, are people who are vision impaired and living with sight loss, as illustrated by the man in the picture who's listening to his phone screen reader, with the text around him reading patient and public involvement and engagement. Our grant funding, which really drives a lot of this work, is divided into 4 key themes, each with significant impact on our activities, to understanding the biology of vision loss and eye disease, prevention, diagnosis, and treatment. So I think it's enough from me. And now it's really time for Gavin to take the centre stage. So I'd like to give Gavin a big welcome and thank him for joining us today from South Carolina. He's an honorary professor as well at the UCL Institute of Ophthalmology, as well as being an associate director of research at Greenwood Genetics Center in South Carolina. And Dr. Arno is particularly interested in identifying the genetic defects leading to rare diseases, particularly inherited eye disease. Over to you, Gavin.
Hi, thank you very much. I hope you weren't expecting me to have an American accent, by the way. I am here in South Carolina, but I was born and raised in London in the UK. So it's nice to be here. It's just the afternoon now, so good afternoon to everyone, and thanks for joining us for this session. You're going to start with some questions for me, is that right?
Yeah. Yeah, so I suppose aside from the welcome, which we've already done, I suppose could you introduce yourself and explain your current role and your previous role, please?
Yeah, sure. So I guess we'll start now and go backwards from there. So I am the Associate Director for Research at Greenwood Genetics Center in South Carolina in the US. So we are a clinical and research institute. We're a non-profit organization, and the aim of our work is to provide genetic testing and genetic care for patients in the state of South Carolina and beyond. Much of the work that we do is on clinical genetic testing for ID, autism, developmental delay, and severe inherited disorders. My role here, or one of the biggest reasons that I was recruited here, is as a director of research for innovation. So the aim is to bring new technologies to advance genetic testing and advance the care for patients here. So that's kind of why I came. And with that, so much of the research that I'm doing now is focused on new technologies, and those are around a technology called long read sequencing, which is hopefully a transformative technology that will push forward clinical diagnostics for patients with heritable diseases, including patients with inherited eye diseases in that. So that's where I am now. I've been here for 6 months now, and my lab is up and running. I've actually got my PhD student from London here at the moment. He's with me for the next 4 months, and I've got my lab set up here. So I also hold an honorary post at the Institute of Ophthalmology, which was mentioned earlier. So I have a research lab based there, and I work closely still with my colleagues at Moorfields Eye Hospital, Professor Andrew Webster, who I think is on the session today, and others in Moorfields. So we still work on genetic data from Moorfields patients by collaboration across the ocean. And I guess that takes me to my previous role. So before I came here, I was the senior scientist for the genetics department at Moorfields Eye Hospital, where all of the genomic testing for patients with genetic eye diseases came through our department, and we helped to analyze and interpret those results for patients and families. I have a research lab in the UK where we were developing new methods of analyzing that data, particularly around the 100,000 Genomes project, which I'm sure we'll talk about in a bit more detail later. to advance the diagnostics in the UK for patients with inherited eye diseases. And then going back a bit before then, so before I became the senior scientist at Moorfields, I held the Fight for Sight Early Career Investigator Award, which was what enabled me to establish my own laboratory at the Institute of Ophthalmology and develop my research program looking for new causes of retinal diseases and characterizing new mechanisms of retinal diseases. And before that, I was a postdoc scientist in the lab of Prof. Webster and Prof. Moore previous to that. And beyond that is ancient history.
Thank you, Gavin. So you mentioned your early career research award that you received from Fight for Sight. So how instrumental do you think that was in terms of really setting up and accelerating your career?
Yeah, so that was awarded in 2017, and it funded. So firstly, it enabled me to establish my own research program. So previous to that, I was a postdoc scientist doing some really exciting work, but I was working on other people's projects and developing my own ideas towards independence. And so that's how the Early Career Investigator Award benefited me. It enabled me to set up my lab and to start exploring those new avenues of research under my own funding and develop those ideas further. And so that award funded my laboratory, at least in part, for 5 years. And during those 5 years, that's when I established myself as a leader in ophthalmic genetics. I could establish a global collaborative network with lots of other labs in Europe, in the States, in Japan, and develop ideas with those and develop collaborative projects. And essentially it really launched my both my academic and my professional career. So it was a huge benefit to me for sure.
Thank you. So you've mentioned in your introduction and when you were explaining your previous roles that you've worked a lot with public initiatives like the 100,000 Genomes Project. How important do you feel these are to advancing research, and how can newer prospective studies like Our Future Health further benefit research? Where do you see the potentials?
Yeah, so I was privileged to work on kind of the precursor to the 100,000 Genomes Project, which was the NIHR Bioresource Rare Disease Study. And this is going back to around about 2013, 2014 when that project was launched. And it was the first of its kind. It was a large-scale whole genome sequencing project to look at the causes of rare diseases, and included in that were around about 700 patients from Moorfields Eye Hospital. So the benefit of working on that for our research and the patients at Moorfields was that several gene discoveries were made from that project. So many new diagnoses were made, many new genes were found that cause retinal dystrophies, and these all fed back into the diagnostic lab and were included from that point onwards in all clinical testing for inherited eye diseases. And that kind of that was a global leading project. That was the first project of its kind to use whole genome sequencing in a diagnostic setting. And from that, really, the 100,000 Genomes Project came about, which was much larger, spanned many more disease areas, and included many more patients with retinal dystrophy and across rare disease. So I think there were somewhere around about 2,000 to 3,000 patients with retinal dystrophy included in that project. And so when we get into these large scale projects that enable these discoveries to be made. Obviously, we can all see the benefit for researchers from that, and we can extrapolate the benefit for patients. I mean, patients are individually getting diagnoses from these projects, but it's also building towards a better diagnostic pipeline for all patients who are subsequently tested. And so we see that in our diagnostic yields over the years. So if you go back to before whole genome sequencing was available at all, we had a diagnostic yield of somewhere around about 40% of patients were getting a positive genetic test back from gene panel testing. And we're now up to somewhere around about 70 to 80% of patients with a retinal dystrophy get a positive genetic test now, due to all of the advances made through these projects and others. And then looking forward to larger scale population-based projects like Our Future Health and using integrating genomic data with public health data. These are going to be transformative for many disease areas, many areas of research in general public health, but also in rare disease as well. And really, we need people to volunteer. We need people to participate in these. Otherwise, they simply won't be big enough to be able to generate meaningful data sets. And so one area we really need help with is to encourage individuals of Black and ethnic minorities to contribute data to these projects, because historically these groups have been poorly represented in not only disease data sets, but in population-based data sets. And so we do worse at understanding the cause of genetic disease in Black and ethnic minorities because of that. So it will be through large-scale studies like this that we will start to better understand the genetic variation across many different populations in the UK and around the world as well.
And to follow on, do you think that might eventually hold hope for treatments or interventions?
Yeah, so I think much of the development of therapies at the moment is around things like gene therapy. So this is a really exciting area of research, and there are lots of different gene targeted therapies in trial at the moment. But as we can understand the genetics, not only the individual genetic cause of disease, but all of the other genetic contributors to that disease, then this will open new pathways to things like genetic modifiers, and not necessarily having to target the individual gene, but being able to target modifiers that will work across many different retinal dystrophies, for example. So these are the things that we can start to think about looking forward to in the future, I think.
Thank you. So you mentioned you've got sort of, in a way, a dual position. You have two labs, one in the UK and one in the US. And how do you think, do you really see that that's important in driving together international collaboration to accelerate outcomes for patients? Or do you think there's still barriers to doing this?
So I think the future, and really the present day, is that we do share data across the globe. So, for example, the 100,000 Genomes Project data, we've just signed an agreement with Genomics England from here, from Greenwood, to allow us to access that data and start interrogating that data. So researchers here will be able to register and access that data now. So we already do collaborate across the world, and international collaboration is essential for driving discoveries. So we are part of a European consortium on retinal dystrophy genetics. We are part of a global consortium on retinal dystrophy genetics. And when it comes to things like gene discovery, now collaboration is key to enable that. So the discoveries that we are making now are exponentially rarer, or they affect very much fewer patients and individuals, broadly speaking. And so to be able to validate and see the real effect of those genes on patient populations, we need to be able to broaden those patient populations to include collaborative groups all over the globe, really. And so, but there are barriers, obviously. There are barriers, particularly between the US and the UK, because the way that genetics are done, the way that clinical genetics are done, are very different. In the US, in the UK, it's all done within the NHS Genomic Medicine Service. In the US, it's very much disjointed. So individual institutes will do their own testing. Data sharing is a bit more complicated, but we work towards doing better there for sure.
Great, that's a good answer. So you've clearly had a brilliant career to date, and you've got many more years ahead of you researching. What would you say is your sort of career highlight to date?
Yeah, it's an interesting question. I think, I can talk about the discoveries that we've made. A gene discovery is a very exciting point in your career, and I've been lucky enough to make several, and to the impact of those discoveries is significant on the knowledge base for rare disease genetics. I guess for me though, it's been a unique experience, a privilege to have been part of these groundbreaking projects like the NIHR Bioresource, which was the first of its kind in the world. And so to be the lead scientist for the retinal dystrophy part of that project was a privilege, and then to go on from there and play a significant role in the Genomics England 100,000 genomes pilot and main study was also a privilege. But I think really the highlight for me is probably now, because we are in a time where genomics is changing so rapidly, and for a scientist to be working now in genomic scientist is the most exciting moment in my career. So the new technology that we are using, things like long read sequencing, Oxford nanopore technologies, sequencing, to be able to answer questions and see things in the human genome that simply were not possible before now is hugely exciting. So I think, if you ask me next year, it might be a different answer, but I think today that's my answer. It's an amazing time now to be a scientist working on the human genome.
Okay, thank you. I think that's proof that you're in an exciting field. So I think we're now going to move on to the Q&As. So please, we'll come on to the questions which have been put into the Q&A function. And we have a couple of free questions that have been sent in, and we've selected some of those to get us started. But please type any questions you'd like to answer by Gavin into the Q&A channel. So the first question is, my father and son are suspected to have choroideremia. However, genetic testing has so far not identified the gene. Are more sophisticated genetic tests likely to be available in the near future?
Yes. So I guess one answer to that is that so currently, although we are doing whole genome sequencing for patients in the UK in the Genomic Medicine Service, the knowledge is a little bit behind in that we don't quite understand very well what's happening outside of the parts of the gene that code for proteins. So a little bit of genetics is that genes are made up of exons and introns. So the exons are the parts of the gene that make the protein. They directly code for the amino acids that put together the CHM, the Rep1 protein. And the introns are gaps between those exons. Now, until the last decade or so, the introns were pretty much ignored completely, and we didn't really understand what they did. We're now starting to be able to understand what the introns mean and what their function is, and understand what variants within those introns do to the gene and how they actually affect the protein. So that knowledge lags behind the technology a little bit, in that we can sequence all of those introns without any problem now, but we can't fully interpret those. So clinical testing currently doesn't look too deeply into those introns because we don't have the ability to interpret what variants may be. So for a gene like choroideremia, or a disease like choroideremia, which is pretty much monogenic, so the vast majority of cases who have a clinical diagnosis, the vast majority of individuals who have a clinical diagnosis of choroideremia should have a variant within that gene somewhere, but it may be a gene that has not been looked at, or it may be a variant that has not been interpreted, or it may be a variant that has not been detected. We don't know the answer to that yet, but as the technology advances, we will better understand those variants. And as we use new technologies like long-read sequencing, which are coming to the clinical labs in the next few years probably, we will be able to detect more of those variants as well. So,
sorry, Gavin, just to clarify for some more perhaps some lay members of the audience, in terms of monogenic, you mean just one gene is responsible? Is that correct?
exactly, yes, yeah. So one gene, one disease. There are a couple of other genes that look similar to choroideremia, with a different inheritance pattern. But broadly speaking, choroideremia is caused by variants in the CHM gene on the X chromosome.
Okay. And when you talk about the non-coding bits of the genome, would this be what some people might have heard of as being referred to as junk DNA?
Yeah, exactly. I mean, that's how it was previously thought of. Now we know it has a function. We know it's important, and we know that variants are there, and some of them lead to dysfunction of the protein made from that gene. So there are variants like that reported in the CHM gene. In fact, one was detected several decades ago when the gene was discovered. And we found several more since then at the Institute of Ophthalmology. So we know they exist. We know that's a mechanism of disease for the CHM gene, but being able to interpret those often needs a functional test. So it needs a second blood sample to be taken from the patient to use for RNA analysis, for example, to be able to understand what the effect of that non-coding variant is.
Okay. So we have another question, which is somebody's question, when you say a positive genetic test, could you explain what that means in lay terms, please?
Yeah, okay. So when we do genetic testing, we find so that the human genome is 3 billion letters long, and we all carry around about 5 million variants in our genes that don't lead to disease. They're non-pathogenic. They don't have a severe effect. And when we do that genetic testing, we focus specifically on the retinal dystrophy genes. We will always find variants in those genes. No genetic test comes back with no variants on it. That's not something that's even possible. But the interpretation of all those variants and finding the individual mutation or variant that is leading to disease is where it becomes very, very complicated. And we now have a method of classifying all variants into 5 different categories. So we go from benign to likely benign to a variant of uncertain significance to likely pathogenic to pathogenic being the strongest category. And for a positive genetic result, the variant or variants need to fall in the likely pathogenic or pathogenic category. And this is based on lots of different pieces of evidence to score those variants in terms of how damaging it is to the gene, how rare it is in the population, whether we've seen it before in other patients and individuals, whether we've seen it in lots and lots of people who are unaffected by disease. So the way that the variants are classified is on the report that patients get from the clinical lab. It will say a likely pathogenic variant has been found in gene X or gene Y, and only those where the variants are thought to fully explain the disease, only those will be considered to be a positive genetic test result.
Okay, thank you. That's helpful. Very clear answer. So we've got another question, which is actually from one of the pre-questions sent in, which is, my son, who's 21, is diagnosed with X-linked RP mutation on RP2. He also has astigmatism. And the person would like to know if there's been any research on the RP2 gene and when we can expect this kind of therapy to begin. And, perhaps a topical question, can we somehow improve damage or fix up the astigmatism to help him see better? And what else could be done to slow down the retinal deterioration besides sitting and waiting for blindness to come?
Yeah, that's a hard question. There are lots of therapies that are being investigated for many different genes, and we know that there are roughly 300 genes known to cause a retinal dystrophy. RP2 is one of the more common ones that we come across in the genetic clinic, and there are therapy investigations ongoing for that gene specifically. It's very hard to predict when specific therapies will make it into clinic and be available for individual patients. And as a molecular geneticist, that's the sort of work that is done by colleagues downstream of my lab. So my lab is concerned with discovery and improving diagnostics. And we work with other groups who are interested in therapeutics, but we don't do that sort of work ourselves. So I'm afraid, it's hard to say when a therapy will be available, but patients and individuals should hopefully be reassured that there are many researchers, and many that are funded by Fight for Sight specifically, who are interested in developing and trying to develop therapies for genes like RP2. And then in terms of what can be done to slow down progression, I think the families are advised well by their clinical teams as to what lifestyle choices they can make, and it very much depends on the specific gene that's causing disease. What sort of lifestyle choices can be made to improve or to slow down progression of disease, and these will be things like to avoid smoking, to avoid going out in very bright sunlight, and don't take vitamin D and all of that sort of thing for patients with RP. But I would, I'm a scientist, I would refer you to your clinical team for the best advice on those things.
Okay. So we've got a question from the Q&A, which is, what do you see as the biggest breakthrough in the inherited eye diseases genetic pipeline in the next 5 years? What do you think is really close?
Well, that's the sort of question I love. I think it's going to be long read sequencing, and the application of multi-omics investigations to genetic disease. So that's a complicated statement.
Yeah.
And and I'll just try and clarify a few of those things. So currently, the way that we do genetic testing is we take the 3 billion letter long genome and we smash it up into tiny pieces that are 150 letters long. We sequence those, and then we try and put it all back together again, and that's really hard to do. The putting it back together again fails for very large regions of the genome, including genes that are very relevant to retinal dystrophy genetics. So RPGR, another cause of X-linked retinopathy, has a region in it that's really hard for us to put it back together again. The opsin array, which causes blue cone monochromacy as well, is very complicated. Now, long read sequencing works differently. So it doesn't smash the genome up at all. It means we can sequence very large chunks of DNA in the millions of base pairs long, the millions of letters long. We can sequence single molecules of DNA in that way. And that means the assembly again is much less complicated. If you compare it, if you imagine it to be like assembling a really complicated, difficult 5,000 piece jigsaw to assembling the same jigsaw that only has 9 pieces, for example. So it's a much easier experiment to do, and it's a new technology that we're working with, and I think it has huge advantages over the current technology, and it will potentially transform the way that genetics research and further down the line diagnostics are done for patients. And then I mentioned multi-omics, which is a broad term that basically means we are able to study genomics, transcriptomics, which is the message of the genes before a protein is made, but study that on a large scale, which is difficult for retinal dystrophies because many of those genes are only functional in the retina. So being able to look at those genes in easily accessible tissues is quite difficult. And then proteinomics, which is looking at the proteins that are made, and metabolomics, looking at the metabolome of the cells affected by disease. So looking at pathways within cells, what's affected, what's upregulated, downregulated, what's functioning and non-functioning in those cells. So I think those things will advance research. The diagnostic impact of those will come very much later. But I think in the next 5 years, for me, long read sequencing will change the way things are done completely.
Thank you. So I suppose a follow-up question would be is about really how important, how will this improve lives, and how important would it be to give patients these genetic and multi-omic answers?
Yeah, so I mean, we're working towards being able to provide a positive genetic result for every patient who is in clinic. That's the aim of my research. And so we improve that by discovering new causes of disease, which is the sorts of experiments we're doing now. Long read sequencing will help us to solve questions and solve problems for patients that can't be solved by the current technology. So, for example, one big problem that we see at clinic in Moorfields is that patients who need family segregation to confirm their genetic result for their report, and I'm sure people who are on the call may have experienced this, when parental samples are needed to be able to confirm a genetic result through phasing, through segregation. Lots of people's families are not available for that. Maybe they're overseas, they may have passed away, there may be patients may have been adopted. That there are lots of reasons why segregation studies can't be done. And one very simple thing that long read sequencing will improve is the fact that we don't need parental samples to phase variants anymore. We can do it all from a single sample. So the availability of that technology coming to the diagnostic labs will mean that the way we approach those experiments will change completely. So that will improve the state of diagnostics, which when we think about therapeutics and treatments for retinal dystrophies, you know, one essential component of that is that patients will need a diagnostic report, a positive genetic test to be able to receive a treatment, whether it be a gene therapy or a gene modifier. So without that, patients who can't get that through the inability to have family segregation done, they will not have access to therapeutics based on that. So that's one way that we aim to be able to improve clinical care, and also, you know, counseling and understanding prognosis and progression of disease relies on that genetic report as well. So these are all things that rely on having a clear genetic report, and that's one area we're aiming to improve.
Okay, so we've got another question that's a bit linked to long read sequencing, which is on long read sequencing. And somebody's put that they would assume that long read sequencing could allow for the identification of some issues that would help in identifying the cause of newer diseases. But beyond that, what benefits might it give to those for which the conditions, I suppose, the diseases where the mutations are already known?
Yeah, so that's another good question. The audience are on their toes, I have to say. So yes, we can identify new causes with long read sequencing. We can sequence parts of the genome that were previously unsequenceable. But another thing that we can do, with bearing in mind the segregation and the phasing that I've just been talking about, is that we can characterize the entire genetic background that the mutations are on. So a mutation on its own may be enough to cause disease, but its severity or how bad the effect of that variant is on the gene may depend on lots of other variants around it. So we call these modifiers. And so we don't understand modifiers very well at all yet, but having the ability to characterize, we call them haplotypes, which is the chunk of DNA that carries all of those different variants on it, the single chromosome that carries all those variants on it. Being able to understand those using a technique like long read sequencing, rather than trying to infer that from short read data, means that we'll have a much better ability to understand the potential modifiers that lie around the pathogenic variant itself.
So they've got a follow-up question, which is, would long read sequencing perhaps help in developing disease models?
Yeah, absolutely. And you know, one example of that is the gene PRPF31, which causes dominant retinitis pigmentosa. It's known that almost half of individuals who are carriers of a mutation in that gene are unaffected. So we call this non-penetrance. So lots of people can be unaffected in families. It can skip generations in families, even though it's a dominant disease gene. Now we think we know where the modifiers of that are, where the variants are that switch that on or off, and they're around that gene. But a technology like long read sequencing will enable us to investigate that to a depth that we haven't been able to do so far.
Okay, thanks. So I think we've got about 5 minutes left for questions. Perhaps we could squeeze in one or two more, depending on the sort of responses. So we've got a question here, which is ophthalmology the next exciting health area in which we can expect to see groundbreaking discoveries? I'm thinking about the things that have come out of oncology, diabetes, and Parkinson's.
So, I mean, for me, it's exciting, yes, and I think, you know, one area where it has proven to be groundbreaking is in the development of gene therapies. So, the first gene therapy was, of course, for the RPE65 gene, which is a retinal dystrophy gene. So, it is leading the way for many areas of research, and I think for genetics, for rare disease genetics, and I haven't mentioned this at all, but one of the reasons why ophthalmic genetics is so exciting for a researcher like me, I'm a molecular geneticist. I'm not an ophthalmologist, but the area of research that I've chosen is retinal dystrophy genetics, because we have a very great understanding of the genetics of retinal dystrophy now, and that's partly due to the fact that, you know, we work with very closely with the clinic, and we have the ability to see the cells that are affected in live patients in clinic, which is a unique feature of retinal dystrophy. You can't see the cells in cardiovascular disease and ask patients what the symptoms are of their disease. So this kind of drives discovery, this ability to combine that clinical data, the patient's biography and all of that stuff with the genome data, drives discovery, and it puts ophthalmic genetics at the forefront of genetics research. So if you look at the 100,000 genomes data results that were reported a few years ago, the pilot study results, the diagnostic yield was highest in ophthalmic genetics. So that's partly due to the work that we did, but partly due to the fact that we have a greater understanding of the causes of retinal dystrophies compared to many other disease areas. So that makes it a great model for future research. So it's a great model for long read sequencing research, which is why I continue to work in this paradigm going forwards. And so discoveries are driven by that fact. So yes, I think it is exciting. I think there are lots of big discoveries to be made, and you know, the next target area is things like modifiers and being able to understand what the genetic background, the effect of the genetic background has on disease. And I think ophthalmic genetics is the perfect sphere for that work as well.
Well, thank you, Gavin. Thank you, and thank you to the audience for all of your questions. I think that's probably all that we have time for today. But we'll try and answer as many as we can after the webinar. So if you'd like further information on either Dr. Arno or Gavin's research, which has been funded by Fight the Sight, or would like to know more about genetics and the eye conditions, and find out more about our research funding, you can do this by looking at these links later on after the webinar's finished. Please don't scramble to write things down, because these will be available to you in your follow-up email, which will include links to the recording, transcript, and this information. So thank you once again for attending, and we hope you found it informative, and we hope to see you at the next webinar in our series.
Thank you very much.
Thank you.